Hello everyone, welcome back to my channel Competitive Botany. In this video, you are going to see APSET Life Sciences previous question paper. The test was conducted April 2024. Okay, so I'm going to give you the question paper with answers. So in the last session of this video, I will also upload Please watch the video till the end. So in this video, you are going to see the questions from 26 to 50 questions. So in the previous session, I have uploaded 1 to 25 questions. So please go through that video too. So let's see the 26th question, list 1 and list 2. Here also you have to match the list 1 with list 2. List 1 consists type of blastula. List 2 consists animals. So celioblastula, steroblastula, discoblastula, periblastula. List 2 consists animals. So here I am going to match the list 1 with list 2. So before that, the answer is, I am going to reveal the answer. So answer is second one. That means celoblastula, type of development seen in Amphioxus. Steroblastula development seen in Nereis. Discoblastula type of development seen in fishes. Periblastula type of development seen in arthropods. So the answer is B. Let's see the next question. Identify the correct statements concerning the enzyme nitrate reductase. Okay, so you have to pick the correct statements among these. First one is, the enzyme is located in cytosol, structurally made up of 4 identical polypeptides. Heme iron is the cofactor for the enzyme, catalyzes the conversion of nitrate to ammonia. So among regarding nitrate reductase, you have to pick the correct options among these. Okay, so I'm going to reveal the answer. So the answer is B. That means, so nitrate reductase is structurally made up of 4 identical polypeptides, as well as heme iron is the cofactor for the enzyme. So these two statements only correct regarding nitrate reductase. Let's see the next question. In fish blood, an increase in proton concentration induces a decrease in hemoglobin oxygen carrying capacity. This is called, options are Bohr effect, root effect, Haldane effect, chloride effect. Okay, so here also I am going to reveal the answer. Answer is root effect. Okay, so let's see the next question. Identify the correct combination of factors among the following that can change gene frequencies in populations. So here 4 statements given. Among these, you have to pick the correct statements, correct combination of statements. Okay, increase in population size, selection, migration, meiotic drive. So you have to arrange them in order. Okay, so I'm going to give you the answer for this also. B is the correct answer. That means selection and then migration and then meiotic drive. Okay, so let's see the next question. Here also you have to match the list 1 with list 2. So first list consists of periods: Cambrian period, Carboniferous period, Ordovician period. List 2 consists organisms. Okay, so I'm going to match the list 1 with list 2. Answer is first one. That means in Cambrian period, sponges are evolved. Carboniferous period, first amphibians appeared. Ordovician period, in Ordovician period, plants are colonized first. Silurian period, corals are appeared. Okay, so for this, the first option is correct one. Let's see the next question. Death of last member in recent times resulted in the total extinction of the species in India. Identify that. Options are Malabar civet, Asiatic cheetah, Chinese pangolin, Great Indian bustard. So I'm going to reveal the answer. Asiatic cheetah is the correct answer. Okay. Let's see the next question. Match the following. Here also you have to match the list 1 with list 2. List 1 consists RNA world hypothesis, metabolism, first hypothesis, neutral theory of molecular evolution, nearly neutral theory of molecular evolution. You have to match them with the appropriate scientist. List 2 consists scientist name. Okay. So I am going to match. First one is the correct one. That means RNA world hypothesis is related to Walter Gilbert, and then metabolism first hypothesis was proposed by Fernando and Rowe. Neutral theory of molecular evolution is proposed by Moto Kimura. Nearly neutral theory of molecular evolution was proposed by Tomoko Ohto. Let's see the next question. Following are some statements to osmotic stress in plants. Which of the following is not correct? Okay, so let's see the options. The accumulation of ions during osmotic adjustment is predominantly restricted to the vacuoles. In order to maintain the water potential equilibrium within the cell, compatible osmolytes accumulate in the cytoplasm. Glucose is one of the most compatible osmolytes involved in osmotic stress in plants. Glycine, betaine, and proline are prominent osmolytes involved in osmotic stress in plants. So, let's see the correct answer. Glucose is one of the most compatible osmolytes involved in osmotic stress in plants is the correct answer. So let's see the next question. Here also you have to match the list 1 with list 2. Equality of means, equality of variances, regression coefficient, correlation coefficient. So we have to match them with list 2. Okay. So equality of means is related to, I am going to reveal the answer first. Fourth one is the, A is the correct answer. So equality of means is represented by capital T, small t. Equality of variances is represented by capital F. Regression coefficient is represented by small b. Correlation coefficient is represented by R, small r. So the correct answer is A is the correct answer. Here also you are going to match the list 1 with list 2. List 1 consists enzymes. List 2 consists values of K-cat for the enzymes. Okay. Enzyme value. Okay. So let's see the matching. Chymotrypsin, lactate dehydrogenase, acetylcholine stearase, catalase. I am going to match the list 1 with list 2. First you have to see the answer. Answer is third one. 3, 4, 2, 1 is the correct answer. That means chymotrypsin is consisting K value 1 into 10 to the power 2. Lactate reductase is consisting K value 1 into 10 to the power 3. Acetylcholine stearase is consisting the K value 1.4 into 10 to the power 4. Catalase consisting the K value 4 into 10 to the power 7 is the correct answer. Let's see the next question. Here also you have to match the list 1 with list 2. Peroxisomes, Balbiani rings, gene battery model, microtubules. So you have to match them with according to appropriate statements. Okay, so let's see the matching. I'm going to reveal the answer. So peroxisomes, so they are consisting glycolytic enzymes. Balbioni rings, they are present in polytene chromosomes. Gene battery model, so proposed by Breton and Davidson. Microtubules, they are present in spindle fibers. So that is the correct one. Let's see the next question. A diploid of E. coli has the lacoperon allelic constitution of IOZY plus by IOZY minus. The production of beta galactosidase and permease enzymes in this cell would be, options are inducible for both the enzymes, inducible for beta galactosidase and constitutive for permease, constitutive for beta galactosidase and inducible for permease, constitutive for both the enzymes. So I'm going to reveal the answer. Third one, constitutive for beta galactosidase and inducible for permease is the correct answer. I'm going to tell you 38th question. SOS, son of sevenless. Okay, the homologue of Drosophila, CDC 25 dash in signal transduction pathway. So options are, is a GTPase activating protein, is a guanine nucleotide exchange factor, is phosphorylated by MEC itself directly activates MAP kinase. So for this the correct answer is, it is a guanine nucleotide exchange factor. Let's see the next question. Arrange the sequence of events in egg sperm fusion of mammals. So options are, four statements are given according to related to egg sperm fusion in mammals. So you have to arrange them in order. Acrosome reaction, sperm lysis whole in zona pellucida, sperm capacitation, sperm binds to zona pellucida. So I'm going to give you the answer. For this the correct answer is C. That means sperm capacitation and then sperm binds to zona pellucida and then acrosome reaction occurs and then sperm lysis hole in zona pellucida occurs. So let's see the next question. 40th question. Here also you have to match the list 1 with list 2. Witch's broom disease in pine, triple response growth in dicot seedling, second internodal splitting in bean, bolting in cabbage. So here phytohormones are given. You have to match them with appropriate answer. So let's see the matching. For this, the correct answer is fourth one. Witch's broom disease in pine is related to zeatin. Triple response growth in dicot seedling is related to ethylene. Second internodal splitting in bean is related to brasinolide. Bolting in cabbage is induced by gibberellic acid. Let's see the next question. To which protein subunit of nicotine acetylcholine receptor binds the acetylcholine to activate the channel at the neuromuscular junction? Options are alpha, beta, gamma, epsilon. You have to pick the correct option. So for this the correct answer is alpha subunit. Okay, so let's see the next one. Genes present on the homologous segments of X and Y chromosomes are known as, options are autosomal genes, Hollandric genes, completely sex-linked genes, incompletely sex-linked genes. For this, the correct answer is incompletely sex-linked genes. Okay, so let's see the next one, which is a bilobed gland in mammals and located just caudal to the larynx and lateral surface of trachea. Options are lungs, thymus, gullet, thyroid. For this, the correct answer is thyroid is the correct answer. Let's see the next question. Phenology of birds deals with. So options are four statements are given: migration, seasonal biological events, heartbeat increase during flight, hy- hyperphosphatemia. Okay, so you have to pick the correct statements about phenology of birds. Okay, so for this the correct answer is 1 and 2, that means migration and seasonal biological events. So they are appropriate according related to phenology of birds. Okay, let's see the next question. Identify the correct statements from the following. Options are: mitochondria evolved from engulfed photosynthetic bacteria. Some of the organellar proteins are encoded by nuclear DNA. Protein synthesis in chloroplast starts by methionine and not by N-formyl methionine. Chloramphenicol is the inhibitor of mitochondrial protein synthesis. Among them, you have to pick the correct statements. Okay, I am going to give you the answer. 2 and 4 statements are correct. That means some of the organellar proteins are encoded by nuclear DNA. That is the correct statement. Chloramphenicol is the inhibitor of mitochondrial protein synthesis. So that is the correct one. Let's see the next question. Which one of the following statements is correct about the homeo box? Options are: it is part of the promoter in eukaryotic genes that code for proteins involved in segmentation. It is a conserved protein structure found in glycolytic pathway enzymes. It is a conserved DNA sequence found in genes that code for proteins which regulate development. It is a conserved protein structure found in tRNA binding proteins. For this, the correct answer is C. That means it is a conserved DNA sequence found in genes that code for proteins which regulate the development is the correct answer. Let's see the next question. For nucleic acid labeling, which of the following radioisotope is employed? Options are H3, C14, S35, P35. Among these, you have to pick the correct one, that is P35 is the correct one, which is used to label nucleic acids. Okay, so in the second order reaction of an enzyme, the rate of product formation is. So here 6 statements are given about the reaction of an enzyme, so you have to pick the correct statements. Okay, let's see the first statement: directly proportional to the substrate concentration, independent of substrate concentration, depends directly on the concentration of two substrates, depends on the square of the concentration of a single substrate, dependent of the coenzyme concentration, independent of the coenzyme concentration. So among these, the correct statements are 3 and 4 are the correct statements. 3 and 4 are the correct statements. Okay, let's see the next question. So as ZDNA shows the following structure. Options are: zigzag course of phosphate backbone, right-handed helix, dinucleotide repeating unit, dimer of molecule 18 angstroms. So you have to pick the correct statements about ZDNA. So for this, the correct answer is 1, 3 and 4 are the correct. Okay, ZDNA is a left-handed helix. You should remember. Okay, so let's see the next question. The type of gene regulation illustrated by lactosoperon in E. coli is, so negative inducer, positive inducer, negative repressor, positive repressor. You have to pick the correct one. It is a negative and inducer type of gene regulation in E. coli. Okay,